יונת יכולה לסגור את הדלת? סגרת את הדלת? סגרת את הדלת? Okay. Hi everybody. So last class we finished the biochemistry of the cell chapter. Just to remind you that we talked about fatty acid. We made a distinction between saturated and unsaturated fatty acid. I told you that the saturation is in regard to hydrogens. So fatty acid that is unsaturated, like this oleic acid over here, doesn't have all the hydrogens that the carbon backbone can hold, so it's called unsaturated. And for example, this palmate or palmitic acid is saturated. We also made a distinction between unsaturated fatty acid between cis and trans. We mentioned the fact that trans is actually a form that is unnatural. So we almost exclusively find it in industrial oils or margarine and stuff like that. And in 99% of the time in nature, you'll find only a cis conformation of this bond. I also showed you like the composition of different food sources that you eat, just to make you comprehend that, for example, also beef fat can have like palmitic and also part oleic acid, so nothing is like absolute. So you don't think that anything is like pure. I told you not to eat margarine once again. Then we talked about phospholipids a little bit. I told you that the way that you will see fatty acids in the cell, in the context of the cell, will not be like free lipids, like I showed you before, like that. Like this fatty acids, they will almost always appear as these phospholipids that can have either 2 or 3 fatty acid chains that are attached to one another with glycerol. And normally this molecule will have one side that will be hydrophobic and one side that will be hydrophilic. And we call this type of molecules that have both these traits amphipathic. So in the cell, normally phospholipids can arrange around themselves or aggregate with one another in 3 major forms. Micelles, which are like single layer phospholipids. Again, imagine that everything surrounding here is water. So the part that interacts with water, the hydrophilic, is pointing outside and the hydrophobic is pointing inward. They can also be stored like as a liposome, which is like a double. It's like a vesicle that has aqueous environment inside and is a double layered. And in most of in all eukaryotic cells, you will find the phospholipid bilayer that separates between the outside of the cell and the inside of the cell, and will create like this impenetrable barrier. We'll talk about it specifically when we talk about in the chapter when we talk about membranes, exactly what does this barrier do for the cell, and what's the function of this barrier for the cell. So we finished by talking a little bit about the types of biologically relevant energies. Specifically, we said that cells can transform one energy to another. I gave the example of plant cells that can transform carbon dioxide and the sun's energy and store that energy in the form of sugar, which all other life forms actually consume. And this is the source. This is like our source of energy. So we are like secondary consumers of the sun's energy. You can look at that. And we talked a little bit about thermodynamic, defining what is exothermic and endothermic, always in relation to the environment. Exothermic is a reaction that will release energy, and endothermic reaction is a reaction that will take in energy. So, for example, we gave the most classic example, which is burning of wood. So you're breaking actually the sugars that we saw that were created before. Again, remember that the wood is composed of a lot of like starch and cellulose molecules that are actually sugars. So the breakdown of these sugars plus oxygen releases energy to the environment. So this is like exothermic reaction. So we mentioned a very important molecule, which is called ATP, and this molecule is actually very similar to a base of DNA, as we will learn, like we mentioned briefly, and we'll talk about it much more in the rest of the semester. And what's special about this molecule is that this molecule is like the energy coin of the cell. Okay, so it has these 3 phosphate groups that are attached to one another, like the phosphate with the oxygen surrounding it. And there's a lot of energy that is stored, a lot of potential energy, you can think about it, that is stored inside these bonds. So the body or the cell uses this energy coin in order to supply the energy to a lot of the reactions that it needs to make. So, for example, if we have a reaction that we want to make, we want to take B plus C, and we want to make substance D, for example, like connect B and C together through a covalent link or something like that, then what will normally happen is that you will have a pre-charging of one of the elements with potential energy, meaning that in this example, substance B will, together with ATP, will be charged with this phosphate, and the ATP will lose its bond energy and pass it to one of the substances of the reaction. Then this charged substance of the reaction will be able to, you can say that the reaction will be charged with potential energy that will help it later do the reaction of B and C equals D. Okay, and the phosphate will be released. Okay, so the breaking of this bond will you can say release or give the energy for the reaction that will produce for this type of reaction. Okay, and this is very common. You will see it. We will see it a lot of times happening in the cell. This type of scheme. Okay, so with that we finished, and we finished this chapter, and we started the chapter on protein structure and function. Again, something I want to say, like a general thing about this chapter, is that it's very hierarchically organized, meaning that in the beginning we talked about the type of folding that a protein can how a protein folds, and in what way do we know that proteins fold. Then we will talk about, after the folding, what type of modification they undergo, the proteins, after they're produced, and finally the degradation of the proteins. We also talk about specific functions of proteins, like some proteins are enzymes, molecular motors, and the mechanism that we know that regulates protein function. And in the end, we'll finish with purifying methods that are in common use when talking about proteins. So yeah, in a second. I'm just doing this is the introductory part. This is just the thing that we already talked about, whatever I'm talking now. So when I finish that, I'll go to the question, and then we'll start. Okay? So, unless you think it's unnecessary, and we can jump.
No, it's fun.
Okay. So, I also think it's fun. Okay. So, we started talking about, I told you that there's this hierarchy, and when we talk about the molecular structure, they have proteins. We said that what is defined, a primary structure is actually the sequence of the amino acid of a protein is defined as its primary structure. There's no folding or anything in the primary structure. But what is important is to know that in the primary structure, for example, we define that there is directionality in proteins. Okay, so a strand of amino acids that are linked together that make a protein will have a directionality from normally from N to C, or if someone says like C terminal or N terminal, then you know it's like one side of the protein and another side of the protein chain. It doesn't mean that in the end, after the folding, that they're going to be in two different sides. So, and we said that a lot of times in proteins you can have as many as 4,000 residues or amino acids in a typical protein in length. Then we defined what are secondary structure. We said that there are three types of secondary structures: alpha helix, beta sheet, and U-shaped turn. And I told you that in the level of secondary structure, we're always talking about interactions that are happening inside the backbone. Okay, we didn't talk at all about the amino acid residues. Like for us, the alpha helix, beta sheet, and U-shaped turn, they're all a result of normally interactions or hydrogen bonds inside the backbone of the inside the backbone of the protein, and we didn't talk at all about the identity of the amino acids. We'll start talking about them when we talk about the tertiary structure and the third structure. So we mentioned the alpha helix. What is unique about the alpha helix is that all the hydrogen bonds in the backbone are occupied. So it means that the identity or whether this alpha helix will be hydrophobic or hydrophilic entirely depends on the identity of the side groups. We mentioned the beta sheet, and the illustration that I showed you before wasn't very good to show the differences between parallel and anti-parallel, but basically you need to imagine that if you have like this is like one protein, and the protein and the backbone is like can go like this and then turn like this and turn like this. So this conformation will be anti-parallel, and this conformation, for example, you can get it if a protein, if the backbone will do some kind of loop. So if it will go like here and then loop like here, then a lot of times you'll get this parallel conformation. And again, it's only relevant when I know that the backbone has directionality. And actually, when you look at them from the side, they look pretty similar. Like in the beta sheets, you always have the residues that are like interchangeably pointed up and down relative to the surface of the beta sheet. But the major difference in our terms is that you see the hydrogen bonds here that stabilize this structure. So here they are not linear, what we call. So there's not a linear line between, for example, the oxygen here and the hydrogen of the amine here, like you see in the antiparallel. And what will this cause? What will be more stable, like this structure or this structure? This one, because we said that the bonds, if they're linear, a lot of times they're also shorter, because there is like less distance between them. You can also see it here, like when you're crossing a boardwalk, you know, Alachson is a son, but that you always need to cross a road directly, because then the distance is shorter. And this will be actually stronger than this conformation. But again, it depends on a lot of different factors. So we also mentioned turns. Specifically, we mentioned glycine and proline, that a lot of time will be located in these turns between like the beta sheets and in general like less structured turns. I showed you the example of the Ramachandran plot, and I was actually asked a question which really bothered me. I'll tell you about it in a second. I told you about the Ramachandran plot and we introduced the terms phi and psi. Phi and psi are the angles that are surrounding. So in relation to each amino acid has a central carbon. That this carbon also has the side groups connected to it and everything else. But so beside the peptide bond that connects between two amino acids, that we said it's a little bit rigid, so it can't move too much, the peptide bond, there are 2 degrees of freedom around the central carbon of an amino acid. Okay, and 1 degree of freedom is defined by psi, and 1 degree of freedom is defined by phi. And we talked about that the fact that there are these two that they can rotate, it doesn't mean that they can rotate in every direction, okay? Or that they're stable in each conformation. And this is because a lot of the steric interference, because again, this is not what happens in reality. I always want you to imagine that in reality you have big electron clouds that are surrounding each one of the atoms, and these electron clouds are actually repulsing or affecting how, like, these angles could be, like I showed you in the model. I'm not going to show you again, but they restrict the possible angles that we can get for fine psi. And this plot actually describes exactly like if you take, I think they took here like 10,000 amino acids out of typical proteins and measured the fine psi angle, and what they got is that most of these angles will be in conformation of either alpha helix beta sheet, and I told you like before U-turn because that's what's written here. But then we talked about specifically about specific amino acids, and I asked you what will be the Ramachandran plot of glycine and proline, and we said that glycine will be like more permissive. It will allow much more angles because it has a very small side group, and proline will be much more restricted because it's actually like one of the angles is degenerated, like it can't actually move because it's the side group is connected to the backbone of the amino acid itself. But then it bothered me, like there was a question here about why isn't there, we said that they're in turns, right? But this is supposed to be a turn, and there's no conformation here. Like it doesn't look like proline can be in a turn, but anyway, everywhere you look, always proline is the typical example of amino acid that you find in turn. So I looked at a different source, and I actually found this plot, which I think is much more accurate than this, like in the description. And actually, like there are also types of turns that can be around this area. You see, like this is type 2 turn, and not all of this area or the conformation of or the combination of the two angles are only beta sheets. You can have like anti-parallel beta sheets, parallel beta sheets, and also type 2 turns around here. And this, for example, can also represent a left-handed turn alpha helix. Most alpha helixes, I didn't mention that, are right turn, and also type 2 turns, but it's less significant. So when we know this, that there is also a type of turn that is in this conformation, then it's more clear why proline is specifically here. So it's exactly where you would expect a type 2 turn. Okay, so now the questions. Okay, because we finished the introductory part. So some of you have submitted these questions to me. This is although it's nice because it shows me that you went over the questions. It's not required. Okay, so there's no requirement for you to answer these questions. I just think that it's a good indication to what are the things that you need to know. Okay, for example, I told you that also a person that doesn't have a background in biology, any background in biology, then I really recommend for you to read the chapters in the book. For example, some of the questions that I will put, we didn't go. It's not going to be like that in the test. Okay, like I'm not going to put in the test questions that are only in the book. I'm not doing that, but sometimes you'll find in these questions that in some questions the answers will also require a little bit of reading in the book. Okay, it's just to test like your general knowledge. So, what cellular functions involve the adenine base? So I will. Is it stabilizing structure? No? Why not?
Yes, I said yes, but I didn't know.
Okay, where does it stabilize?
The DNA, the double helix, is connected by the hydrogen bond to the
Yeah, so this is an example of what I told you before. We said that the adenine base is actually involved in base pairing. We mentioned like in a few seconds the different bases, and I told you that one of them, that two bases connect with one another. And so this is a stabilizing structure specifically in DNA, but we'll learn about a lot of it in following classes. Information storage? Yes, in DNA. Energy storage? Yes, in ATP. So the answer is A plus B plus C. And by the way, also these answers, like these types of questions, will not be in the test. In the test, you have only 4 options. So, following a list of chemical building blocks and macrostructures, match each of the macrostructure with one or more of the building blocks from which it is built. Okay, so the cell membrane, what is the building block that it's built from? Phospholipids, polypeptide, amino acids, DNA strand, nucleic acid, RNA strand, also nucleic acid, and polysaccharides from monosaccharides. Okay? What type of chemical interaction exists between sodium ion and water? Ionic. Okay, I thought it was so there was it's either ionic or hydrophilic actually. Like this was also in the test, and I removed it because it's not very like clear cut, because there's also a hydrophilic interaction. But we will say that this is an ionic interaction. If you remember that I showed you that the water molecules are like hydrating or coating the ion. So we would say that this is an ionic interaction. Monosaccharides different from one another by, I did it really bad. Okay. So, yeah, next time I'll do it better. So, they differ from another by the number of carbons in the molecule. We said that sometimes there are 5 carbons, sometimes 6 carbons. Bond conformation around the carbons. I actually showed you that the differences between a lot of different types of monosaccharides is actually just the conformation of the OH and H. Ring size is different because they can be hexagons or pentamers or pentagons. And the polysaccharides that they form, again, obviously, like the different types of monosaccharides will produce different types of polysaccharides. Okay, so it's all of the above. Okay, is that clear? Questions regarding that? Questions? Okay. Speak now. Okay, so up until now we talked about the primary structure and secondary structure. Now the tertiary structure is much, we would say, much less defined. Like in sense that we can't say that we only have like 3 types of tertiary structures. Tertiary structure is actually the long-term or the long-range folding of the protein, or the 3-dimensional structure of the protein. So, in contrast with the secondary structure, this structure actually really very much depends on the identity of the amino acids. Okay, like the identity of the amino acid will determine in which way the protein will fold. Okay, so. And a lot of time, because this folding will not be very rigid, and the interactions that will induce this folding will be relatively weak, then what you'll get is that, and this is a very important thing to remember, is that the protein, and I have here an illustration of a protein that I made. Okay. So, for example, this is our amino acid sequence, and this will be our primary structure, which is flying from my hands. Okay, so this will be like the level of the primary structure, just a linear sequence. This will be, for example, the secondary structure. You see like the alpha helix that are forming here. And tertiary structure is something much less defined. For example, this can be like the 3D conformation of a protein. And what is important to remember is, because these interactions are relatively weak and not very stable that induce this structure, we call it, like in biology, a lot of time you say that the protein is breathing. Okay, so you need to imagine that molecularly what is happening is that this thing is always moving. Okay, it's like breathing. And this is actually very important for a lot of different functions, and also the regulation of protein. That it, what?
Something you said that
Yeah, unlike the secondary structure, the 3D long-range folding, which is the tertiary structure, strongly depends on the identity of the amino acids themselves, not on the backbone, but on the but on the side groups of the amino acids, or the identity of the amino acids. Okay, so this is an example of different types of different visualizations of a protein. For example, this protein that I have in my hand, and it's important for me to show you them because a lot of times in articles you can see either one of these, and each one of them has a different advantage. So, for example, this is the most simple way to show a protein in 3D, and here you just see the backbone, like just the carbons that connect the backbone. You don't see anything else, but it's a good way to get a nice visualization of how the backbone is going of this protein. Then you have the most elaborate model, which is what we call ball and stick, which actually you can imagine that it's exactly like the model that I showed you here, in which you can see each and every one of the atoms, but you don't see, for example, the electron clouds around them. And so, for example, you can see like that it's much more rich than the backbone from what you saw here. Then you have like the ribbon structure, which is also very common. This is especially good to show secondary structures. So, for example, these are alpha helixes. Beta sheets are like these light blue in the back. But again, you don't see you don't see the residues of the amino acids. And then there's a solvent-accessible surface, which is my favorite visualization. Again, why? Because this is actually how water sees the protein. Okay? So this is how, if you would ask me how a protein looks, this is how it looks. It doesn't look like this, or like this, or like this. Because this is together with the electron clouds. Okay?
These are all for the same molecule?
Yeah. So this is exactly the same molecule. This is a RAS plus GDP, a molecule that's bound to it, which is presented in all in all these different conformations. But it's exactly the same. Here sometimes you see like red and blue, which depicts like areas that are negatively charged and positively charged. But in general, I would say that this is how the protein looks. Okay, and again, it's not rigid; it's moving all the time, a little bit, like expanding and contracting all the time. So, in the level of the three-dimensional long-range structure, what we do have is what is defined protein motifs. So, protein motifs are simple combinations of secondary structures, and in this level, we're starting to talk about function. Okay, up until now, we just said that alpha helix and beta sheet, but we didn't say whether if they have a specific function, and that's because they actually don't really have a specific function. They serve as like a combination of the secondary structure. Is the first level where you start seeing function. So there are hundreds of different motifs that we know, and you can find different motifs in different proteins, and a lot of times the motifs that you will find in a protein will also define the function of the protein. Okay, and I'll give you an example of three such motifs. For example, the helix loop helix motif that you can see here is has a very strong affinity for calcium. So in a lot of proteins that know how to bind calcium, you'll find this motif. And the zinc finger motif, which is like this helix loop, this alpha helix, and then this small beta sheet, and this loop, and specifically also the amino acid, we'll talk about it in a second, that also bind the zinc ion. So these are very good for this is good for calcium binding, and this is good for RNA and DNA binding. So a lot of enzymes and proteins that know how to bind DNA and RNA will have this zinc finger motifs. And the coiled coil motif, for example, is when you have two alpha helixes, that one side of the alpha helix will be hydrophobic, and one side, so the red and the blue will be represent residues of amino acids that are hydrophobic. Okay, they're just depicted here in different colors, so you see it's different. It's from different sides of the helix, like different helixes But what will happen if, because you have these two helices, then because of the hydrophobic effect, these two elements will spontaneously attach to one another or cling to one another. And this is very good, for example, for fibrous protein, where you need to have polymerization of long molecules. Then a lot of time you will have this structure, like this coiled motif that will spontaneously bind to one another. Another thing that we see in these motifs is that we have also consensus sequences. So the identity of the amino acids that we find a lot of times in these areas, in the areas that need to interact with the ion, it's not that you can have any type of amino acid there. For example, here you have to have either aspartic acid, which is D, or asparagine, which is N. So either one of these can be. So how to read this consensus sequence is that you have to have N or D, then a gap, then another N or D, then a gap, then D, N or S, then you have the loop, and then you have 4 other gaps, and then you have to have E or D. Okay. So in order to maintain this motif and this function, it must follow with the rules of the consensus sequence, and you have them for each type of the protein motifs. Okay, so it's not rigid in the sense that you can have either D or N in this position, but for example, the position where you have them is very rigid. If you have, if you won't have a gap, for example, here, then it won't function. This motif. Questions? Okay, so above the level of the motif, we're still in the level of the third structure of the protein. If you have elements that are very large, typically like areas of the protein that are larger than 15,000 daltons, we normally will call them domains. Okay, and the reason that we do that is again because we want to define regions that we know that have specific function or specific shape and structure in order for us to describe better our protein. So, for example, in this protein, we will say that we have a globular domain here and a fibrous domain here. Okay, so it's just like we can say it's names or divisions that help us describe the protein. So, for example, in an article, they can say we targeted the amino acid that is sitting on the surface of the globular domain of hemagglutinin. Okay. And the fibrous domain is shorter in hemagglutinin 1 than relative to hemagglutinin 2. Okay, a lot of times, so sometimes the domain will say something about the structure, and sometimes the domain will say something about the function. So, for example, a lot of times a domain can be something that, in the example of EGF, which is this small protein here, so a lot of proteins sometimes will have EGF. You'll see it in different articles that they will say something like domain, like EGF like domain, which will then tell you that the proteins that they're talking about have components or have regions that look like EGF. For example, in the EGF precursor protein that produces the EGF has a lot of repeats of EGF, and this protein will have this element, then an EGF-like domain and another domain, and TPA will have an EGF domain somewhere else. So a nice way I think it's a nice way to show in metaphor what exactly are motifs and domain if we're talking about protein is if we will now try to define what are the parts of an engine and what are the parts of air conditioning. So we will say, for example, that both of them will have fan motifs. Okay, so that's like small components inside the protein or inside the domain that have the same function. Like this fan and this fan have the same function, but they serve for different purposes. They here they also cool the system here. They also cool the system here, but these are completely different proteins, if you will say. And we don't stop there because the engine by itself is not like a functional protein, and the AC by itself is not a functional entity. But if we combine these two together, then we can say that in the protein car that we have here, then there is an engine domain, and we have a AC domain. And in both of them, you have a fan motif. Okay? So this is like the hierarchy. And it doesn't mean that in other types of proteins, like in a generator, you can have like only one without the other. Or for example, in this greenhouse, you can have only air conditioning without an engine. Okay? So the illustration is okay. I'm not sure if it clears things up. Okay, so up until now, we talked about the primary, secondary, and tertiary structure, but we're always in the domain of the same molecule. And what do we define as the same molecule is that it's covalently linked to one another. Okay, so in my protein over here, so all the amino acids are covalently linked with one another, but a lot of times we have proteins or functional proteins that are made of a combination of different molecules that are interacting together. And this is actually the last level that people will define as a protein, which is quaternary structure. Not all proteins have them. Some proteins will only have like one molecule that will define that protein. But in a lot of proteins, for example, that we'll learn about in neuroscience, they are composed of multiple units. So the most classic example is hemoglobin. Like I don't know if it's the most classic, but it's a very common example. And for example, in hemoglobin, we will say that this is a heterotetramer. So the hemoglobin protein, which is in charge of binding oxygen in our body, will have 4 subunits. So we had, remember in tertiary structure, we had motif, then we have domain. Now we introduce the concept of subunit. When you hear about subunit, it means that it's not the same molecule. Okay? So there are 4 different molecules that together form this mature hemoglobin protein. So we have 2 alpha subunits and 2 beta subunits. And because these 2 subunits are different from another, then we call it a heterotetramer. If they were all the same, we would say that it's a homotetramer, meaning that all of them are made of the same subunits. So in context to neuroscience, it's very common, like almost all receptors that you're going to learn about have this structure, more or less, and this is just an example of the GABA receptor. So the GABA receptor is a heteropentamer. So it has 2 alpha subunits, 2 beta subunits, and 1 gamma subunit. Okay, that you need all of them together, and they all find each other and assemble together to form a functional channel that, in this case, when it attaches to GABA, allows chloride ions to penetrate the cell membrane. Okay? Yeah.
What do you mean they come together? What kind of interactions?
So the interactions that bring them together are the same interactions that we learned about, like hydrophobic effect, hydrogen bonds, electrostatic interactions, and all of these, all the repertoire that we learned about. But I don't know if you remember, we showed like in the end of the last chapter, like that it's really actually hard to imagine how these, and I can tell you that because these are actually different molecules, they're also produced, we didn't talk about it yet, but they're produced from different genes, meaning that they can be not even produced in the same time. So they just are produced, they can be produced like in different times in the cell or different areas of the cell, and then they have to find each other, assemble, and function as a functional protein. So, and the way that they do it is exactly by the fact that the interactions between them are highly specific. So it's like it's kind of a random thing, but it's not really random because biology makes sure that they will be in the right place in the right time together. But then the interaction between them is stable because of the different bonds and electrostatic interaction that we learned about that really fit each other like a key in a lock, you would say. Okay, like these will interact with one another and not with another subunit of a different protein. Okay, so and this is a good place to talk a little bit about protein evolution and define the term of homology. And again, the example, the most classic example is the hemoglobin molecule, or the hemoglobin, not molecule, the hemoglobin protein. And for example, if we're looking back in the evolutionary tree, then we know that there was an ancestral oxygen binding protein that was pretty similar, or probably started by the beta subunit of hemoglobin, or it looked pretty similar to the beta subunit of hemoglobin. And during evolution, it had different changes, and in the end, like almost all living organisms have a capability of binding oxygen, and almost all of them have what we call homologs, or molecules that are similar. And actually, when we talk about homology, they don't have to have really high structural homology. Homology actually specifically says that they have a common ancestor. Okay? And a lot of times they also have structural, they also look pretty similar, and a lot of times they will have also the same function. In this example, the leg hemoglobin here and the myoglobin here will be similar to the beta subunits. And for example, only in the vertebrates, we had this split in the evolutionary tree, which we got the beta and the alpha, and again during the course of evolution, they formed what we call this hemoglobin. Okay, this like hemoglobin molecule, or the hemoglobin protein that will have two beta subunits and two alpha subunits. So, for example, if we think about the ancestral oxygen binding protein, it probably looked more like the beta subunit, and only later during evolution, after this split, then it evolved to be like this heterotetramer that has the ability to bind four oxygen molecules, actually more, and form this structure. Okay, so not only did they split together in evolution, they actually stayed together, but not in the same protein, not in the same molecule, but in the same protein. So a little terminology, and this is also a very nice example of that, about the differences between homology and analogy. So homology is similar due to inheritance or to common ancestor, and analogy is similar in the way that they look. Okay, so a lot of times proteins which are homologous to each other are also similar to one another, so we can say that they're also like analog. But in biological terms, every time you see homology, it's supposed to be, unless they're abusing the term, it's supposed to be traits that are inherited by a common ancestor, and analogy is similarity due to other factors, or due to like evolutionary pressure or other things, but not a common ancestry. Okay, so next class we'll finish with the last level, and then we'll move on. Ken?
What does that last one have to do with Elvis?
With Elvis?
That slide. Yeah. Can you please explain that?
These two look exactly like Elvis.
No. What is that?
The analogy, that they look similar to one another,
Oh.
but they have no common ancestry.
I was trying to have like a deeper meaning or something.
No.
Okay.
It probably has, but I didn't present it. Yeah. זהו, זה כאילו עדיין מגדירים את זה בתור סביונס, כי כאילו ההבחנה היא המקור הגנטי השני, הם שניהם נוצרים מגנים שונים.
אבל יש חלבונים שנגיד, אנחנו לא סופרים אותם,
כן, אז זה כבר נכנס לתחום האפור של ההגדרות, זאת אומרת,
אז זה לא ברור.
כן, אבל זה תלוי נראה לי מה הספרות מגדירה, אם מישהו יגידו שזה סביונס, או אם יגידו שזה דומיין, נגיד במה שאמרתי מ-EGF, כאילו יש פרקורסור ואז זה מפתח, ואז יש דומיין. אז יגידו שזה דומיין ולא סביונס, למרות שזה סביונס. אבל באופן כללי ההגדרה היא כאילו שההבחנה של סטביליות היא חשובה בשביל להבין שזה נוצר מגנים שונים.
אוקיי, גנים שונים ולא מולקולות שונות. עם קשר לסס אומר שזה מולקולות שונות?
כן, כן.
אוקיי.
אבל זהו, הקשר לסס הוא כזה... כן,
אוקיי.
אבל כן.
תודה. Okay, fine. So I was about to not answer your question. Seriously? I'm like, no, thank you. I don't. I want to. Like, come to ask your question. Okay, fine. Ask your question. Then she started. I became very moody after that. Anyways, so then she asked me, yeah, like, what do you want? I said, I have polywood. We already sent an email about polywood. Okay, I know you did, but this is my question. I have a question. We already sent an email. Okay, can you please let me ask this freaking question? What? Show us the biology department. The one on, like, the one that's in the room. She has, like, her the side room. And he walks into the building on the fourth floor, and then to the left is like right, and there's two people that sit in the main room, and then one person sits in the side room. Oh wait, you are yeah. Anyway, so I was like, okay, I ran in, and I said to her, yeah, can you send me someone else? No, he's very nice. Anyway, so I was to say, okay, I understand that you sent an email with my question, and I've seen the email. My question is not about the email. Can I ask it? Okay, what do you want? Okay, so this is my question. I don't know. What's your name? What's your okay? She's she's like, oh, I don't understand either. I'm gonna like talk to her, and then while she's talking, calling her, she's like, you see, this is the reason why you should wait for like you're taking all my time, and I see she has solitaire like on her screen, like from a moment ago where she was playing. And I was like, seriously, you're so annoying. Like, first of all, okay, if you really don't want to tell me, and I'll leave. I will probably leave, but now you're just torturing me. It's really annoying. She's just awful. And of course, like, she emailed me at the end. She was like, yeah, she gave me an answer. I think Naomi is right. Anyway, did I ask her? No. I went to her once over the summer when she, I don't know, we didn't talk for more than like 2 weeks later. I spoke to her for like half an hour, and I was almost pictured in the room. Then she told me that she sent me the email after like 15 minutes.
Okay, so in terms, this is the differences between homology and analogy. We'll maybe discuss a little bit, return to that in the rest of the semester. So the last level, or the last level of what we define the protein hierarchy, is what we call macromolecular assemblies, which is simply a combination of different proteins that will act together just like for a unified goal. For example, one of the more classical examples is the mRNA transcription initiation machinery. This machinery, like, we will learn specifically about that, but it's okay that you don't know what this means. It's actually like this machinery is what transcribes or what converts our DNA to RNA and then later to proteins. So in this transcription, this transcription initiation complex, you have a lot of different types of proteins that are recruited and are working together for this specific goal. But we will not say that this is a protein. Okay, so this is the last level is the last level of the protein, and this level is the level of multiple proteins that are serving a single function. Okay, and these structures can be very large. They can be up to 30 to 300 nanometers in size. I know it sounds small, but it's very large proteins. And for example, one of the more popular and more famous molecular assemblies is the ribosome. So for example, like the machinery or the machine in the cells that translates RNA and produces proteins. So this is an EM microscope, EM EM microscope image, so electron microscope image that is showing a portion of the cell that's called the rough ER, but it doesn't matter. We'll talk about it specifically. And these dots that you see here are actually ribosomes. Okay, so these macromolecular assemblies, or this macromolecular assembly, is so large that we can start seeing it in electron microscopes. So again, to give you a metaphor, I think that the best example that I found is the Power Rangers, obviously, that together, like every one of them, they're different entities, they have different characters, also functions, right? But if there's, in this case, in the case of the Power Rangers, if there's a lot of trouble, then they have to form the Megazord. So they assemble together in order to tackle the situation. So I wouldn't define the Megazord as a protein, it's more like a complex of different proteins. So the next chapter, we'll talk about exactly how this folding occurs, and then what is happening during the life of the protein after the folding. So if we're talking about conditions like theoretical conditions, or what we would like to say like in vitro or in a vial or in a test tube that's isolated, then theoretically there are a to the power of n, and n is like the number of amino acids, different orientations that a protein can fold. So if you imagine like a long if you imagine like this long polypeptide chain, like I told you before, like the primary structure, then because I also told you like in the previous class that the phi and psi angle have more or less like four possible, each one of them has like four possible conformations that they can be, and in general there are like 8 conformations of phi and psi that the protein can fold into, because there are n residues, so it's about 8 to the power of n. And when you think about it, in the end, the protein has to have but what we see actually is that almost all the proteins, and actually all the proteins, will almost, and I say like almost, because a lot of times there are deviations, but they will always fold into the same 3D conformation. Okay, so it's not like you have like after the protein is made, it's this long linear strand, and then one time it will fold like this, and one time it will fold like this. Okay, every time that the protein will fold in the cell, it will have it will fold into the same 3D conformation that is called the native state. And because like I told you also in the past, that the sequences of amino acid, for example, by the way, a lot of the reasons that this native state occurs and we get only this native state are unknown to us. Like a lot of people are studying exactly how this happens, but for a fact we know that it happens, and we think that the interaction with water and the identity of the amino acids and their interaction with water has a lot to do with the reason that you get only the single native state of folding. So there's a theory that's called like this canyon theory of protein folding, or this valley theory of protein folding, that says that again, like I told you before, that the entropy or disorder will always tend to increase. And in these terms, when we talk about this disorder of water, you can imagine that when we have a protein that is in this conformation, then there are hydrogen bonds, there are a lot of interactions that happen with the water molecules surrounding it. Okay? And because of these interactions, the entropy of the system is relatively small. So what happens is that in order to increase the entropy of the system, the entropy of the protein is actually decreased, because the amount of different, or the order of the protein is increased in order to increase the entropy of the system. So in terms of energy, it's said that actually the water, or the interaction with water, is what makes only the native state to be like this more energetically favorable. And the reason that you have this small mini valleys or mini crests inside, is that according to this theory, you can also have like these intermediate stable structures in the middle that are not like the last like native state, that the protein can also be in for a while or for a short period of time. And what helps it, for example, the protein to jump out of this, if the protein folded like not in a proper way and it's like stuck here, so why would it like jump out of this energetically, like because this is more energetically favorable, this is less energetically favorable, and the reason that the protein can theoretically jump out of these values and in the end get down to the most energetically favorable state is again a lot because of this constant movement that I told you about. Okay, so you can't imagine it as to be something rigid. For example, in my protein, if it like folded like that and it's not the proper folding, then because of this movement, what can happen is that eventually it will assume its native state or the right conformation. So, and this is another way to show like the different possibilities. So there actually are the order of the folding doesn't have to be exact, but we do know that very much like what I showed you here, the first structures that are happening are the local structure, like the secondary structures of folding are the first ones that occur, and then you have the long range folding of the tertiary structures, and eventually, even if a protein like starts during this path or starts during this path, meaning that it assumed one type of folding and not the other, in the end they will all assume the same 3D conformation, okay, of the protein. Well, then you will have also difference in the sequence of the amino acid. That's a good question. I would say like everything in biology, I would say that probably yes in some of the cases. And this will have to do with a thing that we'll see in a minute, that in the cell also you don't have only like the cell in some terms it doesn't rely on the spontaneous folding of the protein. It has other proteins that they're called chaperones, and it's a very good name, I think. And these proteins actually promote proper folding of the protein and also catalyze, like make them much faster, because you need the protein also to fold fast, because you don't have time for it to be stuck like in a local valley for a long time. You need the protein to be active. So you want the protein to get folded properly and in the fastest way. So theoretically, because regarding to your question, I could think that if you have different species that have different chaperones, so they might push the folding into one direction and not to the other direction. There might be like a different a difference in that, but in theory, like very generally, a sequence of amino acid will always fold in a biological environment to the same native state. Okay, but I cannot like it might be like different in different species because of these proteins. So the chaperones, there are two types of chaperones, chaperone proteins that we'll learn about. One type is what we call molecular chaperones that bind to the protein and stabilize the folding, and also like you really can think about them like chaperones. They escort the protein during the folding, making sure that it's folding properly. And chaperonins, which we'll learn about also, that facilitates an environment that supports the folding of the proteins. Okay, so counter to what you would think, like chaperonins are very, like according to the name, chaperones are very small proteins, and chaperonins are very large proteins. So it's like the opposite of what you would think. So this is an example of a very famous chaperone protein, which is called heat shock protein 70. Okay, and why is it called heat shock? Can someone think about it? What happens what happens when I boil an egg? Okay, what happens there is the proteins in the egg, they denature, they open, and in the case of the egg, when I cool it down, it stays firm because the proteins cannot denature back. So also in our body, if we encounter like a heat shock or like makat chom or something, and we walk in the desert, what happens is that the proteins start to partially denature. So if the protein was in its 3D conformation, when you apply more heat to the system, you increase the energy, and what actually happens is that the protein starts to like unfold because there's more energy in the system. So this is obviously very dangerous, and this is why our bodies cannot handle more than like 40, 41 degrees. Like the shift in 3 degrees Celsius will kill us because we can't handle the abuse of all our proteins in our body to start to denature because they lose all the function. Okay? Because the function is determined by the 3D structure. So because of that, when our bodies undergo a heat shock, a lot of times you see a very high expression, or the body starts expressing these proteins in very high amounts. Because now you have a lot of proteins that are denatured, and you need to fold them very quickly back to their native state. And this is why they got the name heat shock proteins, because they were discovered by experiment in which, like, I think they were discovered in the fruit fly, that they were exposed to heat, the flies to like mild heat, and then they saw a high expression of these proteins. Because they came as a counter reaction of the insect's body to renature and refold the protein that started to get denatured because of the heat. So this process is, again, like we'll see a lot of times, requires energy, okay, and the energy is in terms of ATP. So after these bind together, they actually before that they bind. So they were like attached to ATP. So the binding itself and the release from the ribosome in the partial folding also when you see like this phosphate group coming out, you know that there was a hydrolysis of an ATP molecule, meaning that it required chemical energy from the cell, okay, in the form of ATP. So this actually happens twice, once here and once when they're released. This also requires energy. And in the end, what you get is a properly folded protein. A different kind of mechanism in which also the heat shock proteins will be involved in. The chaperones will then guide the protein into the chaperonins. The chaperonins are these huge proteins that are made of a lot of subunits. And inside this environment, it's actually like after the protein enters, there's a cap that is like sitting on top of this barrel-like structure, and inside this barrel-like structure there's an environment that is favorable for the protein folding. In a sense, it's like isolates the protein from the environment. It allows it to not only like properly fold, but also makes this process much quicker. Okay, now we have a movie.
In vitro, a denatured protein can refold into its native state on its own. Nonetheless, in vivo protein folding is aided by chaperones in order to increase efficiency and prevent aggregation of misfolded proteins. Molecular chaperones, such as HSP70, assume an open form when bound to ATP. In this form, the chaperones bind nascent polypeptide chains as they're synthesized on ribosomes. The bound ATP is hydrolyzed to ADP. ADP exchange with a new ATP molecule causes the chaperones to switch to a closed conformation, releasing the target protein. A second class of chaperones, the chaperonins, is required to help a small proportion of proteins fold properly. A partially folded or misfolded protein is inserted into the cavity of the chaperonin, where it can fold into its native conformation. ATP binding causes the chaperonin to expand and release the protein properly folded.
No.
So I've already talked.
Okay. So, a few words about what happens when proteins don't fold properly is like all these diseases, basically. Alzheimer's disease, Parkinson's disease, mad cow disease, and this T-rein amyloidosis disease. They all happen when each time like a different type of protein misfolds. For example, the amyloid beta in Alzheimer's and Parkinson, tau protein, which is also in Parkinson and Alzheimer, and the alpha-synuclein, which is also in Parkinson disease. So what actually happens, like, and this is explained by this is a very general like mechanisms, but a lot of time when these proteins do not fold correctly, what happens to them is that they assume like a beta conformation, and beta meaning that they assume like a conformation of a beta sheet. And this conformation, because it has like this large surface area, because of van der Waals interactions, starts to stack, okay, and aggregates. So instead of being like a functional protein, what happens is that the protein starts to aggregate, and this is what you see a lot of time in the cells as amyloid plaques. For example, in Alzheimer, you see amyloid beta plaques. So now you can understand like from where this word or these words are coming from. I must say that it's still unclear in a lot of diseases if this is like a protection mechanisms or toxic mechanisms, but nonetheless, this is what happens when protein misfold. So after the protein is being produced and after it's assumed all its native state and everything is good, it doesn't stop there, because almost all the proteins will undergo a second or additional chemical modifications after their synthesis. So the most common modifications that will happen a lot of time is a chemical modification at one of the sides or one of the ends of the protein, or the carboxylic end or the amine end, or the modification can also happen on the amino acid residues. So these modifications actually really are very critical for the activity, lifespan, and the cellular location of proteins. Okay, for example, a protein can be shipped to a different part of the cell if it underwent a different chemical modification. You can think about these modifications a lot of time as like flags for the cell, like the communication inside the cell, or the way that the cell will know what is the state of the protein, or where it's supposed to be shipped, or what's the activity, or whether it needs to be degraded, will be determined by the modification that the protein will undergo after the production of the protein. And this is just a few examples of very common modifications. The most common one is acetylation, which is basically adding an acetyl group, which is CH3CO, to the N-terminus of the protein. And for example, proteins that are non-acetylated will be rapidly degraded by the cell. So we think that this is kind of a recognition mechanism that, for example, if a protein is present in the cell and it's not acetylated, a lot of times the cell would degrade very fast, maybe because it will be tagged as something that is foreign, that didn't come from the cell itself. Okay, for example, from outside, or it could also mean that the protein was not properly folded, for example. Other types of very common modification is like glycosylation is one of the examples where saccharides can be linked to proteins. You can have again acetylation, like I told you before. Phosphorylation is very common. Adding a phosphate to groups, a lot of times serine, to amino acid groups of the protein can mark this protein or activate a protein also. Like I showed you in the example of ATP, that the phosphate group passes from the ATP to the protein, and it like charges it with energy. So this is phosphorylation. Hydroxylation, where you have an OH group. Methylation is a very famous modification also that has a lot of cellular functions. And carboxylation, and so on and so forth. So in the end, again, it's not important for you to remember exactly what type of modifications can happen. It's just important for you to see like the richness of the differences that can happen to a protein or to amino acids after the protein is produced. So when the protein is produced, it's not the end of the line here. But what we actually see is that I taught you that there are 20 amino acids, for example, but hydroxy proline, for example, is not one of these amino acids. So actually, I'm asking you here, like, how many amino acids are there? Because an amino acid, like, we differentiate between different amino acids because of their molecular structure. And now I tell you that after the protein is produced, these amino acids can undergo modifications. So what it actually gives us is that there's a lot more possibilities of, like, the structure and molecular composition of amino acids because of these modifications, much beyond these 20 amino acids. And actually, this is true in biology in general. Like, this is a very true rule in biology, that when you understand more, you understand that there's much more complexity than you thought before. Okay, like, that's like that in almost every term in molecular biology. We always think that we understand something, and then we see that there is another layer of complexity above what we know. So this is just an example of, like, the increased level of complexity that happens after modification. So other types of modifications can be processing. So for example, peptide segments or parts of the protein can be cleaved out. And this is something that is very typical, for example, in enzymes that are responsible for blood coagulations in our bloodstream, and also program cell death. And finally, a few words about degradation is that, and degradation, or the rate of degradation of the protein, is very important to determine, like, what is the concentration or what is the level of the protein inside the cell, because obviously the amount of protein in the cell is determined by this balance between the rate of production and the rate of degradation. Okay, so degradation is one of the key factors that determines the concentration of certain proteins inside the cells. And there's a huge variability in the lifespan of different proteins. There are proteins that can live, or that their lifespan is like a few minutes, and there are proteins like the opsins in our eyes, for example, the molecules or the proteins that can detect light, that can survive the lifespan of our life. Okay, they can be like, we can be 80 years with the same protein in our eyes. So in general, degradation of extracellular proteins, like proteins that are outside of the cell, and we'll talk about that a lot, so don't worry if it's too fast, but degradation of extracellular proteins takes place in small, like, membrane-bound organelles that we call lysosomes. And in general, cytosolic, or the degradation inside the cell, is largely achieved through a process that's called ubiquitination, okay, or through a protein that's called ubiquitin. And this protein, that won its discoverers a Nobel Prize, which is like Aaron Chekhanov, Amar Eshko, and Irwin Rose, in 2004. They won the Nobel Prize in chemistry for the discovery of ubiquitin, and actually the discovery of the mechanism by which almost, or the majority of proteins end their life. And again, there's a lot of things that are unknown here. For example, how does the cell know when a protein ends its life? We think that there is something that happens during the lifespan of the protein that, for example, it changes its conformation, that's exposing, like, certain sequences on the surface of the protein that might tell the cell that this protein needs to be degraded, or this protein is too old and now it needs to be degraded. So the process of ubiquitination is pretty simple. Again, I don't need to get into all the specific details of what happens here, but what's important to know is that it involves ATP, okay? And there is but what in the end happened in this process is that it's a repetitive process in which the ubiquitin proteins, or the ubiquitin molecules, are repeatedly added to the protein that is bound for degradation, okay? And after there are a lot of residues, or like enough residues of ubiquitin tags on the protein, then it is guided or transported to a structure, or a large protein that's called a proteasome. And this proteasome actually dices up the proteins to small peptides and amino acids. And this is how the cycling finishes. And because this is pretty complex, there's another movie.
So the little pink protein is ubiquitin, and our yellow protein here is ataxin-1. For ubiquitin to be carried to proteins, it needs a carrier. It's called the ubiquitin carrier enzyme. Once our ataxin has done its function and it's ready to be degraded, another enzyme will come called the ligase, ubiquitin ligase. It comes and attached to it. And the ligase will interact with the carrier and take the ubiquitin from the carrier back to our protein. And now it can put more and more ubiquitin molecules.
So the ligase is actually the one that's interesting here, because we have a lot of, actually it said that we have hundreds to thousands of different types of ligases, because this is what actually needs to recognize the different proteins. Okay, so in the question of how the body knows that the protein needs to be degraded, we think that a lot of it has to do with this ligase, because we have different types of ligases that know how to identify specific, like you can say, like stress signals or stress signals on the protein. Like if the protein you can imagine that if my protein, okay, this is a good example. For example, this sequence is a sequence of amino acids. This yellow marking is a sequence of amino acids that the ligase knows how to bind to. Okay, so in the beginning of the protein's life, it was hidden from the ligase. It was in the center, so the ligase couldn't identify it, and the protein did its life. And somewhere along the way, after enough time or something that happened to the protein, then this part got exposed, and then the ligase can attach to it and start the process of ubiquitin tagging, which will eventually
multiple ubiquitin molecules to be degraded. And now, once it's flagged, the proteasome recognizes it, ready for degradation. The proteasome has a cap. The cap opens, the protein unwinds and enters the business end of this proteasome, the catalytic core that chops this protein into little pieces. And this protein will be degraded, and the peptides and amino acids recycled. So this is really what happens for many, many, many cellular proteins.
Okay, so every time you see in an animation that there is something that is too nice, it means that we don't understand. Like it just enters and degrades, and that's it. Okay, so yeah, there's also in this field there's a lot that is unknown about how specifically this happens. Okay, so okay, so a very important class of proteins, which is actually most people think that all proteins are enzymes. So enzymes are specific proteins that promote the chemical alteration of their ligands, or in the case of enzymes, we call them substrates. So you remember when we talked about a chemical reaction? So we can. It's very common to think about the enzymes as being like the chemist of the cells, and what they actually do is that they don't change anything in the chemical equilibrium. For example, like if we put an enzyme inside a vial that knows how to catalyze a chemical reaction, the end concentration that we're going to have, like the concentration of the reaction in the product that we're going to have in the end, is exactly similar to what we would get if there was no enzyme there. The only difference is that the enzyme catalyzes the reaction. Like it makes it happen much faster, and the way that it does it is that it actually lowers like this initiation energy that is needed for a lot of the reactions. For example, you can think about it as adding gasoline to like a fire or to wood. Okay, so the gasoline will be like you need much less energy in order to activate the burn if you have like gasoline there. Okay, so this is actually what they do, is that they create an environment in which the transition state or the energy that is required in order to pass this energetic barrier between the reactant and the product is lower, and thus what happens is that the progression of the reaction is sped up, And this can be very, very dramatic, like in terms of the times that we're talking about in cellular function. So, in addition to their catalytic activity, they're also very specific, meaning that they know how to identify specific conformations of amino acids and specific areas of proteins, and and some enzymes will be like as specific in which that they, even if there's like one hydrogen or one atom that is not in place, that they will identify or not identify this substrate as something that they need to act on. So it's not important. An example, a very classic example that we're going to get into in order to understand exactly what an enzyme does. And in this example, we're going to really dive into what happens molecularly, because again, I think that it's really fascinating, because like I told you like before, it's something that there is no sometimes when you look at it from the side, you don't understand how this reaction can occur, or how this reaction is catalyzed by an enzyme. So I think this example is very important, and specifically this protein, which is called protein kinase A, or pKa, is a very famous protein. The function of this protein is actually to phosphorylate other proteins. So it transfers a phosphate group from, so this is a model of this pKa. It has two areas. It has the large domain and small domain. You see, domains can have also stupid names, but they help us like define the protein. So and we have this is the active site is between the large domain and the small domain, and you also have a glycine lead, because there is a glycine amino acid there. It's called like a glycine lead. So here you see it like assembled, meaning that you have the target peptide, or the part. So this is not the entire protein. You're just seeing like a small fragment of the protein that the enzymes knows how to bind to. And you also see here inside this cleft a nucleotide binding pocket. In this case, it's ATP. Okay, so nucleotide is like ATP is a type of nucleotide. So in this nucleotide binding pocket, you'll find ATP. And again, the reaction that this enzyme catalyzes, and every time you will see an enzyme that's called kinase, it actually means that this enzyme phosphorylates some other protein. And phosphorylate means that it has to add a phosphate, normally from ATP, but there are exceptions that it's not ATP, but normally it transfers the phosphate from ATP to the target peptide, and this in turn normally activates this target peptide. It's like I told you before, you need to think about it like it's charging the target peptide with energy, and this energy will help the protein or the peptide to do its function later on. So actually I talked about this. Okay. Yeah. Okay. So what actually happens here is that now we're like zooming into the different areas of into the area or the catalytic site of this enzyme. So if we have our ATP molecule here with these two phosphates, everything else but this like the ATP and this blue thing here, everything else that you see here, these charges or this lysine or asparagine that you see like these numbers next to, they're all part of the enzyme. Okay, so this is the ATP with the three phosphates. This is the target peptide or target protein that has a serine residue that's sticking out. All the other elements that you see here are part of the enzyme. So, and the reason that they have numbers next to is actually the number. If we're looking at it's numbers that represent the location of the amino acids relative to the primary structure of the protein. Okay, so for example, if this is the first amino acid of the protein, and this is the last amino acid of the protein, so and there's like 100 amino acids here. So and if in the middle I have serine, then this serine will be called serine 50. And if over here I have another serine, then this serine will be serine 75. Okay, so this is just how we define these size groups. And the reason that we see these size groups is that these size groups, in what they do is that they create an environment in which there's like a tug of war that is happening here. So specifically, the positive charges, one of the magnesium ion that is bound here, and this lysine, and in the other side, the magnesium that is in this side, are like pulling away the negative charge of the oxygens. Okay, so they're like taking away the negative charge of the oxygen. Thus, what they're doing is that they're making the intermediate state in which the oxygen is like the oxygen of the serine will now try to like what we call chemically attack this phosphate. And because the condition that it had before was that these oxygens were like surrounding the phosphate and creating a very strong negative charge around it, which would actually repel the charge of the oxygen. What is happening into the core of the enzyme is that these negative charges are being cancelled out in order for the oxygen to come in close proximity to the phosphate. And what will happen in the next step is that this oxygen is going to steal the phosphate from the ATP and turn it into what we call adenine diphosphate and not adenine triphosphate, like it was before. So you see, like, the enzyme is actually creating an environment that is lowering the amount of energy or increasing the probability of the oxygen to be bound to this phosphate. Thus, it catalyzes this reaction. Okay? And you can read about it later. So, a lot of times, because in the cell we have enzyme, we see a lot of reactions in which they're ordered one after the other in terms like you can think about it like a factory, that each machine comes after, like, uses the reactants or uses the reactants of or uses the products of the reaction that they had before as reactants for the next reaction. So in the cell, much like in a factory, we also see this, that enzymes that are linked together in the process, in the materials that they form, will be in close proximity to one another. And also sometimes they will really interact with each other, meaning like they will form like something that is similar to what we saw in the assembly of different subunits, for example. So different enzymes will know how to interact with one another in order to be in close proximity and form like a complex that will know how to convert these reactants to these products. Because again, when they're in close proximity, it also means that statistically the reaction, like the reactants that are needed, for example, for B, will come right out of A and immediately move into B, into enzyme B, in order to be produced into the products of B. What we also see sometimes in evolution is that like during the course of evolution, we don't know exactly why, and this is like also a very general example. Sometimes they also fuse together to be like, if we saw enzymes that during the early courses of evolution were different proteins, sometimes during the course of evolution they actually fuse together and form like a single protein or a single protein that has different subunits that assume the functions that that beforehand were separate into different proteins. Okay, so I think before I start talking about molecular motors, because I have a lot to say about that, I think we'll stop here, also because of my voice. And we'll continue next class. We're going to finish this chapter. We still have to talk about molecular motors and regulation of proteins, and then we'll talk about these methods. And we'll already move to the next chapter after. Okay, so once again, you don't have questions, or actually you do have questions. I think in this chapter I divided the question into two. So you do have questions, and I'll post them after class, and that's it. If you have questions, I'll be happy to answer. אותה